For decades, the bedrock of cardiovascular prevention has been the standard physical examination and the lipid panel. But inside the body, this diagnostic framework frequently misses the actual moment of biological failure, where a pulsating blood vessel suddenly destabilizes and ruptures. Half of all heart attack victims present with entirely normal cholesterol levels at the exact moment their heart fails. A standard lipid panel tracks the transport of fats through the blood, but it fails to measure the mechanical durability of the vessel walls themselves. To maintain that structural integrity, the body relies on omega-3 fatty acids, specifically eicosapentaenoic acid, or EPA, and docosahexaenoic acid, or DHA. EPA and DHA are essential polyunsaturated fats that form the flexible architecture of cell membranes. When doctors do recognize the need for these fats, the standard clinical response is to prescribe generic omega-3 supplementation, usually without any prior blood tests to verify the patient's actual cellular deficiency. True cardiovascular risk depends on whether the individual cells of the heart and arteries are physically equipped to handle biological stress. Many patients take these supplements daily, yet the active ingredients often fail to integrate into the target tissues. Instead, the standard fish oil capsule dissolves in the stomach, and the liquid inside darkens and breaks down before it can reach the cell membrane. A major clinical review published in 2018 looked at this exact phenomenon and concluded that standard omega-3 supplementation frequently produces no meaningful physiological change in the patient's body. The missing pharmacological metric determining whether an oil actually works is the Oil Stability Index, or OSI. The OSI measures how long a lipid can resist oxidation and remain structurally intact when exposed to stress. Because the medical community largely ignores lipid stability, millions of patients consume highly unstable, rapidly oxidizing supplements that provide almost no cellular benefit. This creates a compounding failure in modern treatment. Clinics are monitoring serum markers that fluctuate daily, while attempting to treat the underlying inflammation with the wrong formulation of oxidized oils. To actually deliver EPA and DHA into a human cell, the formulation requires a biomimetic approach. A biomimetic approach is a design strategy that mimics the natural chemical protections found in living marine ecosystems to safely transport the lipid. The bioavailability paradox is resolved once the stability of the delivery vehicle is prioritized over the concentration of the raw nutrient. This animation shows the baseline of cellular health, the physical fluidity of the phospholipid bilayer. The cell membrane must remain highly flexible for transport proteins to properly open and close. Inside this membrane, a structural competition is constantly occurring. Omega-3 molecules provide elasticity, while omega-6 molecules, like arachidonic acid, provide rigidity and trigger inflammatory signaling. For a human cell to function properly, the biological optimum is a 3-to-1 ratio of omega-6 to omega-3. However, modern epidemiological data shows the average global diet has pushed this ratio to as high as 35-to-1. As omega-6 dominates, the membrane physically thickens and stiffens. This physical rigidity blocks the cell's transport proteins, causing essential nutrients to literally bounce off the hardened exterior instead of being absorbed. Simultaneously, this exact same mechanical failure traps the cell's own metabolic waste inside, preventing toxins from being flushed out. When waste builds up inside millions of rigid cells, it creates chronic subclinical inflammation. This is a sustained, low-level immune response triggered by localized cellular entrapment. As this localized membrane failure spreads across trillions of cells, it escalates into systemic tissue damage, eventually causing entire organs to fail. Maintaining cellular fluidity is the primary mechanical requirement for long-term physiological health. Systemic chronic disease is the macro expression of a micromechanical failure in the cell membrane. When marine oils are extracted from their natural matrix in a fish, they lose their inherent biological protections, leaving them chemically vulnerable. This diagram illustrates lipid oxidation. When exposed to oxygen, light, and temperature changes, EPA and DHA molecules structurally degrade, shedding reactive particles known as free radicals. Clinically, this degradation is rancidity, an accumulation of oxidized byproducts that trigger aggressive immune responses. The body identifies these compounds as threats and initiates a rejection process, preventing the rancid lipids from integrating into the membrane. This creates a very heavy metabolic cost. Instead of repairing the cell, the body must expend vital energy to hunt down, neutralize, and dispose of these supposedly therapeutic free radicals. This rapid chemical failure explains exactly why patients taking standard fish oil capsules see absolutely no improvement in their cellular omega-6 to omega-3 ratio. Unprotected omega-3 is biochemically indistinguishable from a cellular toxin. The solution to this chemical roadblock is biomimetic formulation. This requires rebinding the fragile EPA and DHA molecules with natural lipophilic antioxidants before they enter the body. The most effective chemical shield for this process comes from polyphenols. Polyphenols are powerful micronutrients extracted from pre-harvest extra virgin olive oil. By combining wild-caught fish oil with high polyphenol olive oil, along with vitamin D3 and vitamin E, the formulation creates a synergistic blend. This polyphenol shield locks the omega-3 molecules in a stable state, preventing oxidation while the oil sits on the shelf, and protecting it as it travels through the harsh environment of the digestive tract. This chart visualizes the resulting nutrient density. A single 12 milliliter dose of this synergistic liquid provides the exact same EPA, DHA, and polyphenol content as 18 standard fish oil capsules. This stability and cellular absorption rate is maintained whether the formulation is delivered as a raw liquid or bound into a microencapsulated powder. The data in this published study shows that participants consuming this polyphenol-bound formulation experienced successfully reduced blood pressure, alongside a definitive increase in blood omega-3 levels. The researchers also noted a distinct physiological trend. Participants who entered the study with higher starting blood pressure experienced the most significant clinical benefit from the oil and powder. Achieving consistent clinical outcomes requires shifting away from isolated nutrients and utilizing synergetic, food-based matrices. To accurately measure this structural change, we have to look past standard lipid panels and focus on the erythrocyte, or red blood cell. As these cells are actively navigating through the vasculature, they are continuously absorbing and integrating available dietary lipids directly into their cell membranes. The lifespan of a red blood cell is a strict biological constant of exactly 120 days. Because of this specific life cycle, extracting and analyzing the erythrocyte membrane gives clinicians a perfect, unalterable moving average of the patient's cellular nutrition over the previous 4 months. The result of this analysis is the omega-3 index. The omega-3 index is a direct measurement of the EPA and DHA levels physically built into the red blood cell membrane. Short-term blood tests reflecting yesterday's meals are diagnostic noise. The 120-day red blood cell index is the true signal of long-term cardiovascular health. The clinical weight of this metric is validated by the Framingham Heart Study, a multi-generational cardiovascular research project that has tracked patient data since 1948. This comparison chart plots predictive success rates from the Framingham data. Standard cholesterol panels failed to predict a single major health outcome out of 5. In contrast, the omega-3 index successfully predicted 4 out of the 5 outcomes. By simply measuring membrane lipid ratios, researchers could accurately predict a patient's risk for sudden cardiac death, heart disease, stroke, and total mortality. The data revealed that patients who maintain an omega-3 index above 8% experience a 90% reduction in their risk of sudden cardiac death. This establishes a clear clinical roadmap. The goal is to move a patient from the high-risk category, which is an index below 4%, into the optimal zone, above 8%, over a single 120-day cellular cycle. The protocol is a strict 3-step process. Test the baseline index, saturate the body with polyphenol-bound lipids, and retest after the cellular life cycle is complete to prove the structural change. By shifting focus from fluctuating serum levels to the 120-day life cycle of the cell, cardiovascular health becomes a measurable, predictable outcome of cellular nutrition.